We're now going to talk about the different stages of protein purification. So if you have a set of cells, you've expressed a protein, or it's a target organ in an animal, or a set of E. coli bacteria that you've grown, or a yeast, what's the first thing you have to do? You've got to break the cell open, and it turns out that sometimes that's not so easy. You can use homogenization, as shown in the experiment to the right, or you can treat with an enzyme, lysozyme, which is an eggs, is able to break the cell membrane in bacteria and open them up. It's actually a defense in the egg against invasion by bacteria. And once you've broken the cells, you need to separate the cellular contents. There's an opportunity to fractionate, to divide the cellular contents into specific components. And one way of doing that is centrifugation, and we'll talk about that more in just a second. In bacteria, when you break them open, what you have is the cell debris and the cytoplasm. So there's not much to fractionate. You just spin out the cell debris and you have your soluble fraction that you work with. But in complex cells, in eukaryotic cells, it's necessary to fractionate into cellular components. And centrifugation, as you see in the picture on the lower right, is the way that that's done. So here is how you would go about centrifuging a eukaryotic cell extract. You have your tissue cells, and you've homogenized now, and the cells are broken open. If you spin them in differential centrifugation, you put them in a centrifugal field so that there is a force on the components that are in the solution. You will find that if you spin them at 800 G for about 10 minutes, then you get a pellet that has the nuclei and the cellular debris, and everything else remains in the supernatant. So you can take that supernatant and put it in another tube, keeping your nuclei, and you can take this tube, spin it for 15 minutes at 20,000 G, and you get a pellet that's enriched in the mitochondria and in chloroplasts if you're dealing with plants. Now you can keep that pellet, and you can take the supernatant, and you can spin it now at 100,000 G. Note that we're really escalating the force, and you may even need to move to a different type of centrifuge as you go through this process, because some centrifuges can do the low G separations, but you may need an ultracentrifuge for the high G separations. If you take your supernatant and spin it for 60 minutes, an hour, at 100,000 G, that's 100,000 times the force of gravity, then what you get is a pellet that's enriched in the microsomes. And microsomes are parts of the plasma membrane and the cell's internal membranes that have sort of come together and enclosed so that they're small pockets of material. And if you leave the microsomes behind and you take the supernatant and you spin that at 150,000 G for 3 hours, then you can get a pellet that's enriched in ribosomes. So if you know where your protein is, or can test where your protein is across this spectrum, then it's possible to pick the fraction that contains your protein, and that's the first step in your purification, is this sort of crude separation. Once you've done that, there are some other crude methods that you can use, and one of them is precipitation. And these crude methods can be useful, but they're not going to give you purity. Well, let me back up about that. Occasionally, you'll be really lucky and you can use a crude method and get your protein pure, but it's rare. So one way to precipitate is to use the fact that at the isoelectric point of the protein, that is when the protein has zero charge, it is least soluble. And you might think for just a minute about why, when it has zero charge, is it least soluble. And just put that in the back of your mind. So you can make a protein come out of solution if you place it at its isoelectric point. More commonly, salts or temperature are used. Salts are useful. Ammonium sulfate in particular is a very useful precipitant, and you can use different percentages of ammonium sulfate to fractionate. So you can look at the test tubes in the upper right, and you can see that the solution gets more and more turbid, indicating more and more precipitation. And that's exactly how it will look with ammonium sulfate. You can also use temperature. Now, most proteins, as we've talked about, are not likely to be temperature stable. How many non-covalent bonds hold proteins together? 1 or 2. Maybe if you're lucky, 3. And so they're not going to be very heat stable. But some proteins are heat stable. Everybody else precipitates out, and the protein that you want will be enriched in the solution. And occasionally you'll have a protein that's cold stable, and some proteins are not cold stable. But mostly these are crude methods. If you're using this sort of precipitation method, you have to separate the precipitate from the soluble proteins by centrifugation. And then you have to figure out, where's your protein? Is it in the supernatant or it is in the pellet? And then you can take it to the next step. You can also do a series of precipitations where you start with low ammonium sulfate and take out some of the proteins, and your protein is soluble. And then you take out your protein and you leave the proteins that are still not precipitated. So you can use this as a crude method, but a method that gives you fractionation. So now that you've kind of cleaned it up a bit, if you will, the next step is chromatographic methods. And the name implies what it is. That is that this method was developed early on with molecules that had color. They were chromatic. You could see where the proteins were, and hemoglobin's a great example. If you're purifying hemoglobin, you can see exactly where it is because of the red color. And not all methods allow you to see that, but of course the illustrations here are going to be colored so you can see what's going on. And it turns out that there are a wide variety of matrix materials that you can use. Some of these are charged, so they separate based on the charge of the protein. And the charge of the matrix can either be positive or negative, and so if a negatively charged matrix will exchange positive ions. A positively charged matrix will interact with and exchange negatively charged ions. So the proteins are negatively charged, the matrix is positively charged because you're using ionic interactions. And the more positively charged a protein is with a negatively charged matrix, the tighter it will bind. So this type of ion exchange separates based on the charge of the protein. And an anion exchange column will exchange anions, but it will be positively charged, and vice versa. A cation exchange column will exchange positively charged ions and will be negatively charged. Another example is a gel filtration or molecular sieve column. And what this is a column that contains balls, little tiny balls that have channels in them. And the size of those channels determines whether a molecule can enter the ball or not. Little molecules, relative to the size of the pores in these particles, will enter into all of the particles that form the column. Large molecules can't enter inside, and so they will move much more quickly. And so this separates by size and shape, where larger things come out first, they go along the outside of the particles, whereas small things enter the particles and come out slowly. And finally, well, there are 2 more things. Hydrophobic is an apolar surface on the column, and it will separate by surface apolar character. You might think for a minute about what kinds of proteins would have surface apolar character and might be separated using a hydrophobic column. And finally, you can have a specific target molecule. Let's say that something binds to penicillin. You're trying to isolate penicillin binding proteins. And so you can link penicillin to your column and then pull out anything that binds to penicillin. So you're using the affinity or the ability to bind to whatever target you've immobilized, and those are called affinity columns. So we have ion exchange chromatography, which separates by charge, anion exchange, negative charge on the protein, positively charged matrix. Cation exchange, you're exchanging. There it's moving back and forth. It's the positive charge on the protein and a negatively charged matrix. In gel filtration, you have separation by size and shape, and the smaller proteins or molecules are able to enter into your gel particles. They take longer to come out than the larger proteins, and intermediate ones are just intermediate. So normally there is a column. These columns can be short, fat ones, like even 3 or 4 inches and 10 inches long. They can be really narrow, thin ones, a centimeter by 100 centimeters. It depends on what kind of column you're doing and what you're doing. They're now high pressure columns where that can be used under pressure, where the process goes more quickly. Basically, for all of these, you apply a sample and then you apply buffer solution continuously to the top of the column that basically moves your sample through the column. So behind your sample is a buffer solution, sometimes a gradient. You may increase the salt concentration over a period of time or over a volume, but you have a buffer solution that's applied, and you collect the proteins that are you collect all these fractions. The test tubes at the bottom would be called a fraction, and you collect the fractions that will contain the eluted and hopefully purified proteins. So it turns out that if you just use these general chromatographic methods, you can get pretty good purification. You can use the crude methods, combine them with chromatographic methods, and that was what we used for decades in purifying proteins. And you can get reasonable purification by this approach. But it turns out we've gotten smarter, and affinity methods have been developed where you can get the greatest specificity in purification. So basically what you attach to the beads in the column is a component that is able to interact with the protein specifically. For example, you have an antibody that recognizes the protein that you want to purify, and you immobilize that to the column. Or you can have a tag that you've added onto your protein using DNA methods. And for example, a histidine sequence added to your protein has the capacity to bind very well to nickel, and you immobilize nickel on your column. So the combination of tags and methods, and there are multiple combinations, can give purified proteins. So if you immobilize, for example, an antigen in an attempt to purify an antibody, or an antibody to purify an antigen, you can flip this around. Or you put nickel on your column supports where the pink balls indicate the inert matrix to which whatever your protein binds is attached. Then if you're trying to get an antibody specific for the blue antigen, then you can see that it binds. If you have a mixture of antibodies and you have other antibodies that have different specificity, they will not bind and will pass through the column, and any non-antibody proteins will pass through the column. Alternatively, if you have nickel attached to the column support and you have a histidine tagged protein, a His6 protein, so that you have 6 histidines in a row, the histidines will interact with the nickel, and any proteins that do not have this His tag will pass through the column, and the protein that is tagged with the histidine sequence will bind to the column. And you can elute that, get it to come off with increasing concentrations of imidazole, which mimics histidine. So there are a whole variety of combinations that can be used for purifying proteins, but the tools that we have available today are much more robust than historically, where we just had a few types of columns that you had to use, and you had to use combinations of those and different ones in sequence in order to get purification. So if you knew that you had a protein that was much larger than other proteins, which type of purification would you choose for that protein? If you had a protein that is known to bind strongly to a positively charged partner, what type of purification? What's the nature of the column that you would use to purify that protein?
Proteins can be purified